Good afternoon, everyone, and welcome to Axiom's actual second town hall post ASCO. We just wrapped actually RCC, and this focus will be specifically within prostate cancer. For those who are attending via LinkedIn, we also have it broadcasting on YouTube as well. And there was also kind of attendees on Zoom. So we're on multi-platforms here. So on the next slide, I'd like to kindly introduce Dr. Sartor from Tulane University, as well as Dr. Agarwal from the University of California, San Francisco. And during today's discussion, they'll be going over two kind of key broad topics based on data that was presented at ASCO, which just wrapped up earlier in the week. Beyond kind of some introductory opening remarks, the main portion of today's discussion is going to be around that of nuclear medicine. So some of the key data coming out of the VISION trial, along with kind of the role of PSMA theranostics, as well as from a diagnostic lens. Additionally, they'll delve into some of the insights from real-world evidence and kind of then provide some closing remarks. For those who are attending, please submit your questions through the Q&A button. Should be at the bottom of your screen, and we'll do our best to address everyone's questions. All right, I'll pass it off to you, Dr. Sartor and Agarwal.
Maybe I'll kick it off with a little discussion of the VISION trial, and perhaps there's a little bit of pride of authorship, but I think this might have been the most important of the prostate cancer presentations. It was a plenary. Dr. Michael Morris was the one who gave the presentation. I happened to be senior author on the abstract and co-PI on the study. The VISION trial took a look at PSMA 617, which is the molecule, and it's tagged with lutetium-177, which is the isotope, in patients with metastatic castrate-resistant prostate cancer. Next slide. If we look at this group of patients, you're going to see very quickly that they were very far advanced. In order to be eligible, you had to have metastatic CRPC. You had to have prior treatment with at least one androgen axis inhibitor, and that would be abiraterone, enzalutamide, apalutamide, those types of drugs. You could have more than one, and many of the patients, in fact, did have more than one androgen receptor axis inhibitor. You had to have a minimum of one taxane, but you could have had two. And about 38% of the patients had prior cabazitaxel. Now, all the patients were going to receive what we call protocol-permitted standard of care. And protocol-permitted standard of care was given in combination with the PSMA lutetium-177. So we had to ensure that the combination between whatever standard of care was used was going to be safe with the PSMA lutetium. As a consequence, we excluded chemotherapy and radium-223. Now, immunotherapy, we're not so much afraid of, but the truth is there was only one immunotherapy that could be appropriate for this type of patient, and that would have been the pembrolizumab for those with mismatch repair. If you had a mismatch repair, you should definitely be treated off protocol. Please note that at the time we started, the PARP inhibitors were considered to be investigational, so the PARPs were excluded. But anybody who was requiring of chemotherapy, anybody who investigator thought could benefit from a PARP inhibitor or from pembrolizumab or from radium, those patients could certainly be treated, but just not part of the protocol. So we educated the investigators about what was appropriate, what was not appropriate on the protocol, and that if an investigator felt that chemotherapy was appropriate, then they should go on chemotherapy. But it turns out that everyone had had one prior taxane, 38% had cabazitaxel on top of the docetaxel, and the vast majority of patients who were screened were either deemed to be unfit or refused further chemotherapy. ECOG performance status 0 to 2, life expectancy greater than 6 months. Now, the patients were selected for PSMA positivity And the PSMA positivity on PET scan was using gallium-68 PSMA-11. What was positivity? You had to have positive metastases greater than the liver, and then we had negative findings on a cross-sectional imaging. If you had a PSMA negative metastases in a viscera of 1 centimeter or more, you were excluded, or in lymph nodes of greater than 2.5 centimeters. So patients with large lesions or PSMA negative were not considered to be eligible. Now, it turns out about 87% were. So going to the actual treatment regimen, randomization was 2-to-1, protocol permitted standard of care for everybody, PSMA lutetium, 200 millicuries or 7.4 gigabecquerels every 6 weeks. 4 cycles were planned, but if the investigator felt as though there were benefit, then they could increase up to 6 cycles. CAT scans and bone scans were done every 8 weeks during treatment, every 12 weeks during follow-up, and they was subject to a blinded independent central review. Now, there are 2 endpoints. There are alternate endpoints: overall survival and radiographic progression-free survival. Next slide. One of the things that happened is there was tremendous enthusiasm for the study. And early on, if patients were not randomized to the PSMA lutetium, there's a strong tendency to drop out. Well, that's unacceptable in a prospective randomized trial. What did we have to do? We had to take strong measures to diminish the dropout. The first thing we did was sites that were flagrantly not keeping their patients on trial were deleted from the study. So we stopped accrual at certain sites, then re-educated the investigators about the importance of following everybody, which seems to be investigator 101. But I think the enthusiasm for being treated with this drug was such that there was disappointment if they were not randomized to receive the lutetium. Bottom line is, we ended up having to do a reset. And the reset included replacing the patients that initially were dropping out. The trial was initially powered for 750 patients. We took it up to 831. And then we had to do an RPFS analysis on a subset of 581 patients. So you'll see us going back and forth between the OS and the RPFS, but it turns out it doesn't make that much difference. Next slide. Primary endpoint, overall survival. Intent to treat analysis. All randomized patients, all 831. And what you see is a hazard ratio of 0.62. Median was prolonged from 11.3 months in the control arm to 15.3. I think all of us wanted more, but the median of 15.3 versus 11.3 is unequivocally positive, with a p-value less than 0.001, and these patients truly benefited. Many of them benefited even more than what you can see here. Next slide. This is the RPFS. Remember, this is an analytic subset of 581 patients, but you can see, again, a hazard ratio of 0.4, highly statistically significant, median 8.7 versus 3.4 months. So more than a doubling of the RPFS. So this combined with the overall survival unequivocally leads us to conclude that this is an active regimen. Now, not everybody does particularly well. Look at the top of that curve, and you can see even at the first set of scans that about 20% of people have rapid progression. So not everybody is going to benefit. Some patients will have rapid progression. But the median number of cycles was 5, and many of the patients received 6. Almost over 40% of the patients received 6 cycles. Next slide. What about adverse events? Well, you can see that there were more adverse events of all grades seen within the PSMA lutetium arm. However, let me point out something important that's not shown here. The standard of care arm was examined for adverse events for only 2 months, because that's how long they stayed on trial. The PSMA lutetium arm, these individuals were observed for 7.5 months. Not quite 4 times longer, but a much, much longer period of time. So certain of these adverse events may be in part related simply to the disproportionate period of follow-up. Nevertheless, there's more fatigue, there's some bone marrow suppression. If you get to the grade 3-4, you can see on the right-hand side, and not really grade 5, but leukopenia 2.5%, grade 3-4, anemia 12.9, thrombocytopenia 7.9. Dry mouth is part of the therapeutic side effect of this agent. You end up radiating salivary glands 39.3%. Nausea, vomiting was higher than I would have expected, 39.3, almost all nausea, not much vomiting. Renal effects I don't really think are related. I think it's just a longer period of follow-up. Same thing true with secondary primary malignancies, and intracranial hemorrhage was pretty much balanced between the two arms. Next slide. When we look at the secondary endpoints that included objective response rates, and here we see complete responses in about 9% of the patients on the PSMA lutetium. 41.8% of the patients had partial responses. So CR plus PR is a little over 50%. That certainly is quite good and very different than the control arm. Next slide. We'll look at PSA decline rates. And again, you can see a waterfall plot here. Please note that these are confirmed declines. Some of the other studies with PSMA lutetium just used best decline. They didn't use confirmed decrease. So when you begin to look at these percentage of PSA declines, 46%, greater than 50%, that's a little bit different than some of the other studies, but the other studies did not use confirmed decrease. So you can see quite clearly that 46% had confirmed decrease of PSA, 50% or more, and compared to the control arm, 7%. Next slide. Conclusions. PSMA 617, and the 617 is important. Remember, this is a very particular small molecule. Tagged to lutetium-177 is safely combinable with standard of care. It typically included hormonal treatments, abiraterone, enzalutamide, bisphosphonates, external beam radiation, glucocorticoids, etc. Safely combinable standard of care with lutetium extends overall survival and delays radiographic disease progression. As a whole, is well tolerated. Only about 5% of patients actually had dose reductions because of toxicity. And we believe that these findings will warrant adoption of PSMA lutetium as a new treatment option. And this, of course, will be up to regulators throughout the world. But I believe that the regulators will look favorably on these results given the strong overall survival benefit. So that concludes my presentation on the VISION study. Rahul, I don't know if you had questions, take questions from the audience. Glad to help out as best I can here.
Yeah, no, absolutely. Thank you, Dr. Sartor. I think there's one question that came in from the audience. I think this is kind of always a big debate in prostate cancer when it comes to sequencing patients. So with regards to lutetium-177, what patient type do you see this in? Will this be before chemo? Will this be after chemo? And how might you start to think about this from a sequencing perspective? Will it go, we have PARPs in play as well. Be kind of curious to hear both of your perspectives around, once in clinical practice, what type of patient will you treat with this agent?
Well, I personally think that the label will follow the inclusion criteria requiring patients to receive prior abiraterone, enzalutamide, apalutamide, darolutamide, one of these novel androgen axis agents, and a taxane. So I think that the label will dictate the practice, and more trials will be necessary in order to move it up. Now, those new trials are underway, PSMA Edition
Mhm.
The PSMA-4 trials are using the PSMA 617 lutetium-177, but it'll take more trials to change the patient population relative to that chosen envision, in my estimation. Rahul, would you agree?
I would agree with that. I mean, I think we have to follow what the data say currently. And currently, this is the only phase 3 result we have with lutetium, and they clearly were in the post-chemotherapy setting. And so I think agree with Dr. Sartor that I think that the initial label across the various regulatory agencies will be in the post-chemotherapy setting. I think there's a strong biologic argument to make to move lutetium earlier. We do know that in heavily pretreated prostate cancers, some cancers do lose PSMA expression, and we do see a fair amount of heterogeneity of PSMA uptake on the diagnostic PET. The definition of what constitutes a positive scan in which to treat someone with lutetium has also varied across a number of the studies. This study chose a fairly liberal definition such that there was only a screen failure rate of about 12%, I believe, of patients who did not qualify in a heavily pretreated. So with a more stringent definition, perhaps that percentage would have been higher. This has been seen with the studies done in Peter Mac in Australia, where they use a combined PSMA plus FDG PET selection strategy. There, the screen failure rate is closer to 30%. So clearly there are some prostate cancers that lose some PSMA expression. So I think there is a rationale to move earlier. As Dr. Sartor said, there's multiple phase 3 studies underway in the pre-chemotherapy mCRPC setting. And there's also planned studies in the metastatic hormone sensitive setting as well. So I think it'll be interesting to see the results. It will take several years to get there. But it may be at the end of the day that the lutetium is used more widely pre-chemotherapy. I would say on the patient side, there's certainly a lot of preference or desire to delay or avoid the need for chemotherapy, despite the phase 3 data that we have with docetaxel and cabazitaxel. So I think there's, from a patient perspective, also driving that preference or desire to treat pre-chemotherapy.
Yeah, you bring up an interesting. Go ahead, Dr. Sartor, please.
Well, I was going to say, what Rahul's raising an interesting point. If you look throughout the United States at patients who die from prostate cancer, it turns out that no more than 60% of the patients ever receive chemotherapy. And it may be considerably lower than that because actually it's hard to get population-based estimates. Estimates are easy to derive from the larger centers, which are probably more prone to give all the available therapies. If you go to a place like the UK, it may be only 20-25%. So many patients never get chemotherapy. This trial has in part been criticized because there was not chemotherapy in the control arm. But guess what? If you look at second-line chemotherapy across the board, probably at most 20-25% of patients ever get a second-line chemotherapy. So if you'd required chemotherapy to be given in the control arm, you would have required patients to consent for a therapy that most of them don't want. So the way it was designed was to create a big, broad population. Rahul noted that a fairly liberal inclusion criteria for PSMA scanning, uptake greater than liver. And if you go to the Australian studies, much more patients were excluded. We were actually surprised, or I was surprised about that 12 or 13% patients were excluded. I was expecting it to be higher because of the known heterogeneity. But the results are what they are. And to me, it was interesting and surprising. This liberal inclusion criteria, 87% of patients, 87 and a fraction percent of patients, means that a lot of patients will be eligible for this once they fail the chemotherapy and androgen receptor pathway. So I think this is going to be available to a lot of patients going forward.
That's great. Yeah, actually, you addressed one of the questions I was going to ask around the comparator arm. We've seen a lot of chat around that of like, why wasn't it compared against cabazitaxel? Is that kind of potentially harmful on looking at the strength of the data? But the point I'd kind of like to bring up with both of you, and I know we'll be talking about this a little later on, we obviously recently saw the approval of Progenix PYL agent.
Yep.
Telix is in the process of developing their gallium-68. Will there be a need for some form of PSMA confirmation from the diagnostic end upon approval, or kind of that? What's your expectation for that once in the market?
I'm actually thinking that PSMA imaging will be treated as a predictive biomarker, just like we do genetics in order to define populations for the PARP inhibitors. And we have to find a BRCA1, BRCA2, and then put the patient on because those patients benefit. I think it's likely that there'll be a PSMA PET requirement. But given the, that 87, 88% of patients, it's even conceivable that the FDA could just say, okay, treat everybody. I don't think that will occur because we used it in the inclusion, but because so many of the patients, in fact, were PSMA positive. I think, in fact, if you'd taken an unselected patient population here, the study would still have been positive. I mean, after all, the hazard ratio was 0.62 for survival. That's strong. Yeah. So I don't know. We're going to see what the regulators say. It won't be up to me. But I know this. I know that the vast majority of patients in this subset would have fit the inclusion criteria because we demonstrated that to be true.
Yeah, I think in the post-chemotherapy setting, I think that makes a lot of sense based on this trial, a very high screen positive rate. It does raise the question whether it really needs to be a companion diagnostic as part of the label. My sense is that I would agree that I think it's likely to be included just because that's the way the study was designed. On the other hand, I think in the pre-chemotherapy studies where the bar for efficacy will be higher and the comparator arm will have more active life-prolonging therapy, and knowing that there still could be heterogeneity in that setting, depending on the definition that's chosen for PSMA PET, one would imagine wanting to hang on to that predictive marker just because it would be likely to enhance the magnitude of benefit for those studies. So I think it'll be interesting to see how the data play out.
Yeah, no, absolutely. I think one question, obviously, with the entry of radiopharmaceuticals is how might this change kind of your practice engagement from the multidisciplinary perspective? With the role of the oncologist, bringing in kind of the radiation oncologist, how are those dynamics, do you think, going to play out? And do you think it'll kind of potentially inhibit access to the agent, depending upon the type of setting the physician's treating in?
I'll jump out and maybe make a controversial statement, but so many patients want this therapy. I think the doctors are going to adapt. I think if you don't offer this therapy, that they're going to leave the practice. And I'm seeing that right now. I've got it open as an expanded access. I am absolutely snowed over with patient requests. Chicago, Montana, people are coming out of the woodwork because they want to be treated with this agent. And after the FDA approval, there's going to be a lot of patients who demand this therapy. And if your facility doesn't offer it, well, then they'll go find another one that does. That's the way I'm thinking about it. Now, that's a controversial statement. I don't know. I mean, Rahul, what are your thoughts?
I 100% agree. In the short term, when this is the only lutetium agent approved and it's, you have centers that have expanded access open, still limited availability, there's this tremendous pressure that we face on the treatment site to really accommodate as many patients as possible. But I 100% agree, you have to be able to offer it. I do think moving forward, it is an interesting paradigm in terms of the collaboration that's required between nuclear medicine, medical oncology, radiation oncology. I think Mary Ellen Taplin had a really nice, in her discussion after the plenary presentation by Mike Morris, just that this is going to require a little bit more education and collaboration in how we manage and treat patients with lutetium. At our institution, we're planning to embed one or several nuclear medicine physicians within our medical oncology group. They will see these patients during the course of their lutetium, but it's really meant to be a multidisciplinary type of model to really, maximize care, because there are certain parts of expertise that the nuclear medicine physician can offer that we are not trained, or our background is not that as a medical oncologist. I think it'll be as the drug rolls out, that multidisciplinary collaboration will be important.
Absolutely. No, this is great. And I think what would be nice is kind of continuing this discussion, right? Obviously, kind of, I think in the next slide, Evan, what we'll see is Dr. Agarwal is going to walk us through some of the data when we think about lutetium-177 in combination with pembrolizumab.
Yeah, great. No, I'm happy to go through this abstract. This was a study that we ran here at UCSF, so I'm certainly not an unbiased commenter on this. This is my abstract that I presented as a poster, and it's I would just say as a broader comment, we're likely to see many different, of course, lutetium combinations, lutetium plus immunotherapy, lutetium plus PARP inhibitors, so lutetium plus ADT in the hormone sensitive setting. So there's likely to be many different combinations and exciting work to try to understand how we can enhance PSMA expression to boost response. So, I view this as kind of really a very preliminary initial foray. But to just set the stage of what we were trying to do, this was really a phase 1B type study, really signal finding type study. So not a randomized practice changing type of trial, but really to get preliminary data. Very different structure and design from the phase 3 study of Vision study. So this was a single dose of lutetium, not repeated doses. So single dose of lutetium in conjunction with continued checkpoint inhibitor pembrolizumab until progression. And unlike Vision, these were pre-chemotherapy MCRPC patients. One of the other differences is that definition of PSMA PET positivity. So we use the same gallium PSMA 11 diagnostic scan, but our initial definition that we used required 3 or more PET-avid lesions, not 1, but used a comparator of mediastinal blood pool initially. We've since amended that for the part 2 of the study to compare it to background liver. This just sort of gives you a sense of there's really not, one airtight definition of PSMA PET positive scans. We did not do a paired FDG PET, part of the study to keep it streamlined. And so this is what we really report on as the part A of our study. It was really 18 patients, so small number, but we really trying to investigate 6 patients per schedule. And you can see the schedule is outlined there. It really pertains to when that dose of lutetium is given in relation to when pembrolizumab was started. So schedule 1, lutetium is given first, followed by pembrolizumab. Schedule 2, they're given concomitantly on cycle 1, day 1. And then schedule 3, the lutetium is actually given after the start of pembrolizumab. And we enrolled 6 patients per schedule, so a total of 18. We've since started enrollment in Part B and really chose Schedule 1 to take forward as our sort of quote unquote recommended phase 2 dose and schedule. And accrual has been going well. We expect to be finished with Part B accrual by the end of this year. And so there's, as Oliver was saying, there's just a lot of patient interest. And so we really try hard to accommodate as many patients as we can. In terms of the results, if we go to the next slide here. So these are the baseline characteristics of the patients. You can see the 18 patients listed there. Prior ASI therapy was required. You can see that a quarter of the patients had both agents. Actually, a little bit less than half of the patients had both Abi and enzalutamide. Because we required measurable disease by soft tissue RECIST criteria, this inherently enriches for maybe a little bit more aggressive patient population. So you can see that nearly 40% of the patients had visceral metastases. So a little bit sicker patient population than probably the average pre-chemotherapy MCRPC patient, just by virtue of requiring the measurable disease. Importantly, we did try to get baseline biopsies to characterize somatic genomic profiling as well as germline testing. You can see that we had 1 patient out of the 18 with an HRD mutation, 15 were wild type, and 2 that were still in the process of collecting status. No patients with MSI high tumors were enrolled. So really a patient population that you would expect with pembrolizumab single agent from Keynote 199 and other studies. We know the response rate with pembro alone in this unselected patient population is somewhere between 5 to 10%. So it gives us a good historical control. In terms of the safety and tolerability, it's very well tolerated. Keep in mind, we're only giving a single dose of lutetium. So we don't really expect, and this is a pre-chemotherapy setting. So we're not necessarily expecting the same degree of bone marrow suppression as we saw with the Vision study. And in fact, it's been very clean from a hematologic toxicity profile. You can see really just very mild grade 1 type heme tox, but And then in terms of immune-related side effects, we had one patient who had an inflammatory grade 3 arthritis with schedule 2, but really no other dose-limiting immune-related adverse effects. And so essentially the way the study was structured was to have these modified DLT type definition, and essentially all 3 dose schedules were tolerable. We didn't really run into any DLTs other than that one grade 3 arthritis. This just shows some of the preliminary efficacy data, and I think that you see on the left the PSA waterfall plot, and on the right the swimmer's plot in terms of showing the patient's duration of treatment. We also label the patients who had an objective response by RECIST criteria, and then patients in terms of duration of therapy and what reason they came off study for. So first, just looking at the right figure in terms of the swimmer's plot, I think clearly we see, we had 8 out of 18 patients with an objective response. So this is a little over 40%. But encouragingly, we're seeing this subgroup of 5 patients that had a fairly durable objective response, given that they only received a single dose of lutetium. So I think it's definitely intriguing. It's small numbers. I certainly wouldn't make too much of this, but as a hypothesis generating type of data, I think it is intriguing that you can give a single dose of lutetium followed by pembro in an unselected patient population and see these durations of response carry out. So I think we're pretty excited about seeing these 5 longer term responders. The actual response rate in PSA decline is probably on par with what you would expect with lutetium. I think it's really that subset with extended duration of response that we're trying to dive into in terms of the correlatives and really understand what's going on. We built in pretty rigorous correlative studies looking at paired biopsies pre and post lutetium, which we're going to be doing a pretty deep dive and trying to understand the immune microenvironment. The premise being is the lutetium somehow priming the immune microenvironment to make it a quote unquote more hot tumor that's going to respond to pembrolizumab. Next slide. This just shows the RECIST waterfall plot as well. You can see the majority of patients did have at least some regression in some of their target lesions, and then 8 out of the 18 which met the 30% criteria for a partial response. We didn't see any complete responses on the study, so you can see that the max suppression we got was just shy of 80%. However, again, the durability was fairly impressive for a subset of those 5 patients. This patient on the right was that patient who was the longest on treatment on the swimmer's plot that I showed you on the prior slide. And so this is a patient that had prior both abiraterone and enzalutamide. Nothing in his genomic profile that necessarily would make you think would be an exceptional responder to pembrolizumab, and actually was on study with a durable partial response for right around 18, 19 months before he finally progressed. So really a pretty durable response, and has us encouraged. We're gathering more data in the part B of our study, which is ongoing.
Mm-hmm.
So this is just a conclusion slide. I think that we're, again, I would highlight early, this is only 18 patients, but encouraging in terms of seeing these subset with durable response. I think the pieces that we need to fill in still are really the biology and the correlatives. What are we seeing in the tumor? Is there something that the lutetium is doing to really change the dynamics of what's going on in the microenvironment? So between the paired biopsies and some of the peripheral blood assays that we're actually in the process of analyzing right now, I think that'll hopefully give us a good clue as to what's going on biologically with lutetium. We know that with external beam radiation, you can see a potential favorable abscopal or priming effect, at least in a subset of tumors, but I wouldn't say that that's a very robust effect that we see. And so hopefully with more targeted systemic radioligand therapy, we can further enhance the ability for these tumors to respond to immune checkpoint inhibition. And then we should have the additional 25 patients in Part B enrolled, hopefully by the end of this year. And then sometime next year should have the aggregate clinical data to be able to present.
So one of the things that I might ask is imaging the immune response.
Yep.
Or at least the T cells. You could have imaged CD8 or imaged CD3 cells and had a little better idea about what was going immunologically. I do worry just a tad about tumor sampling, because simply there really is so much heterogeneity. And when you look at a needle biopsy, you're just taking just a microcosm of the potential whole. So I might ask that image-based biomarkers involving the immune system be brought into play if feasible.
Yeah, it's a great comment. We actually do have a nuclear medicine a radiochemist in UCSF that's we're planning on bringing on a novel PET imaging probe that's actually a granzyme B PET imaging probe that actually does measure sort of the immune activation. So that we're hoping to bring on during part B and do the pre-post imaging. But I think it's a great point. You know, we definitely think that you need to sample across tumors as best you can. Hopefully the peripheral blood analyses will also give us some sense of that isn't limited to a single tumor for sampling, but you're spot on. And I think we're hoping to bring that online soon.
And then one other comment, you know, you might have a little pride of authorship here, but as a pretty objective observer, I leave that 44% objective response rate and single dose. I'm thinking that's a little bit interesting. Now, you did select the patients a little bit tighter, and they were pre-chemotherapy, but nevertheless, that's a pretty nice response rate for a single dose of lutetium. We know absolutely that you cannot get that type of response rate with Pembro in unselected patients. So it could be something going on here. I'm intrigued, and congratulations for doing some good work.
Yeah, no, I appreciate that. Yeah, no, we're definitely intrigued as well. I think, you know, it's just preliminary, but we're certainly interested.
Yeah. Dr. Agarwal, I'd be curious to kind of hear your perspectives on, you know, do you think there's room, I think to your point, Dr. Sartor, as well, for 2 doses? You know, like what was the decision to go with 1 versus 2 in part of the protocol design?
Yeah, so I mean, there is an analogous study underway with Peter Mack in Australia that I think is 3 doses of lutetium plus pembrolizumab, and I don't think there's been data presented yet from that study. I think one of the concerns we had with repeated doses is, do you start to run more of a negative effect, and do you start to ablate different components of the immune response? And so we have some internal data with pembrolizumab plus external beam radiation that suggests you may induce some of the myelosuppressive cells within the microenvironment with radiation. And then you certainly can get ablation of different components of the immune response, perhaps with repeated doses, with lymphopenia and some of the sort of hematologic side effects you get with repeated lutetium. I don't think there's one right answer we know yet. I think we really wanted to make this study a proof of concept. If we can give a single dose and get as much relatives built in pre-post to really build the argument that, hey, something favorable is going on here. This isn't a simple just add on 2 therapies, but is there actual biologic sense or synergy that we're actually creating here? I think it was important for us to design the study with a single dose. One of the intriguing things we're sort of thinking about is, rather than sort of a fixed schedule of lutetium, you know, does this kind of duration of response we're seeing with some patients who then subsequently progress open the door to a little bit more of a sort of treatment as needed type of schedule with lutetium? Meaning, you know, can you extend the duration of time between lutetium doses with the pembrolizumab, but opt to design a study where you have a second or maybe a third dose at the time of progression after initial response? So there's a lot that could potentially go into it. I think it depends a little bit on, you know, what happens with lutetium as a single agent in the pre-chemotherapy setting. You know, if you were to think about taking this concept forward to more of a larger, you know, registrational type of design, I think there's still some work to be done to try to understand what the combination dosing schedule should look like.
And also be kind of curious to understand, you know, there's been a couple of questions of this come up in the chat of, beyond a checkpoint inhibitor such as pembrolizumab, are there any other mechanisms that you think might be interesting to potentially combine with lutetium?
Well, I think there's a lot that biologically makes sense. We know that, you know, if you even look at things like hormonal therapy, like androgen deprivation therapy, we know there's an inverse relationship, and it's complicated, but there is this inverse process where you can potentially induce PSMA expression early on with the initiation of ADT. And so those studies are underway in the hormone sensitive setting. Clearly, because this is a form of beta particle radiation, there's a role for evaluating with other DNA damaging agents such as PARP inhibitors. I do worry about the hematologic toxicity if you sort of start to combine these agents and what patients' bone marrow reserve will look like. But clearly there are a lot of interesting combinations that can be approached. Oliver, I don't know what your thoughts are on combination therapy.
No, I think you nailed it. I think the only issue you might have if you try to move this forward is you might have more interest from a Merck than you might have from a Novartis, because
Yeah.
you don't really give a whole lot of the lutetium.
Exactly.
But I understand your proof of concept. But I like the idea. I made the point of maybe some image-based assessments of immune function, which I think could be intriguing. And I think you're already thinking about that. And so I think that's great. Other combinations of DNA and in the DNA repair is something that if you inhibit makes intuitive sense for something like the lutetium, which damages DNA. So PARP inhibitors, I mean, you can say that with your eyes closed. And, there are other ones out there. We have ATR inhibitors now. There are DNA PK inhibitors that are under development. So there could be some interesting work with inhibiting DNA damage repair. And those are the ones that seem intuitively obvious to me. This, and as well as the need, I think, to explore more with the PD-1, PD-L1 axis. So, these are the obvious ones. I'm sure there are going to be others. Hormonal therapy synergizes with radiation, but yet there's an upregulation. For instance, if you add enzalutamide, you upregulate the PSMA expression if you're looking at the castrate resistant setting. Yet, when you kill cells, you might end up with smaller tumors. And some people would say that when you don't have the same degree of crossfire, you have smaller tumors with a beta emitter, such as lutetium, that you may not have as good cell kill. This sort of begs the question, should we be going with alphas? And, there's a whole lot of data now developing on the alpha particle front, targeted alpha therapy, and that becomes a whole new discussion. So you better watch out. We'll spend all of our time talking about two abstracts. Maybe we need to go see a few more. But there's a lot of implications here. A lot of good work to follow.
Yeah, and kind of in thinking about that as well, and making sure we save time for kind of going beyond nuclear medicine. I might suggest that we had two kind of core presentations on the diagnostic front, and I believe Dr. Agarwal is going to kind of give us a brief overview, but it might be kind of interesting as we move to the next slide to even start thinking about some of the clinical implications here. And I think kind of obviously, I'm sure many of our community physician colleagues are interested in some of the logistics end and kind of what that means from their day-to-day practice.
Yeah, sure. And I can be pretty concise with these. I think this was a study. The strength of this study was that it was in some ways mirrors the prior study with gallium PSMA that was done at UCSF and UCLA that led to FDA approval of our imaging agent back in December of last year. But the nice thing about this study, it was a much larger prospective. I think there was 15 centers. And so the strength of it is really showing the feasibility across different centers that you can image patients in the recurrent setting. So if we go to the next slide. So they took patients with biochemical recurrence, either those after prior surgery or radiation treatment. And the take home for me was really the methodology that this was 17 centers in 15 countries. So international study, many different radiologists ordering and completing these scans. And as you show in a second, you still see high quality data in terms of lesion detection. So we go to the next slide. So 65% of the patients essentially had a positive scan at the time of biochemical recurrence. And no surprise, you're more likely to have a positive scan if you had a higher Gleason score, a higher PSA at the time of the PET, shorter PSA doubling time, and then prior radiation as the primary therapy. And they did build in a change of management endpoint. I think we've seen a number of these studies that have this endpoint. It's certainly interesting to look at. I would view it as a pretty soft endpoint. It's certainly not the same as looking at actual clinical outcomes. But nevertheless, the number 56.8% is in line with what we see with other studies, about 65% of patients with a change in management based on PET scan, which is quite astounding to see that. Really, it's just complete sea change in how we think about biochemical recurrent prostate cancer with the introduction of PSMA PET. And then importantly, the last bullet point there, consistent imaging results across many different countries, many different radiologists. So I think this gets to your question, John, about just the real world applicability. I think it's very clear that the inter-reader rate variability is actually fairly low with PSMA PET, and you get pretty high diagnostic quality across many centers. Yeah, I think
we can go on to the Condor study.
The conclusion. And then maybe quickly, I'll just talk about Condor. This is really more a subset analysis of their prior phase 3 registrational study. So this is the DTF-POIL imaging agent. The main contrast here is with the 18 fluoro compounds. Of course, they don't necessarily need to be synthesized on site. And so when we think about real world applicability and synthesizing the cyclotron and shipping out to radiology centers, this should dramatically enhance the feasibility of PSMA PET imaging across a number of different, imaging centers, certainly here in the United States. Next. So what this study is doing, this just shows the schema. These are patients who had a biochemical recurrent prostate cancer. This just shows you the chemistry of the compound. These bind to PSMA with high avidity. But essentially, what the crux of it is how do you define the truth? When you see a positive lesion on PET, many times these lesions are too small to biopsy. And so now a number of these registrational studies have looked at a composite. And so we look at biopsy. If it's there, great. If it's not, then we look at regular imaging, including Axiom or fluciclovine PET. And then if it's not there, then you look at what's the PSA change with radiation to those PET-avid lesions. And that composite truth has really been now widely adopted across a number of studies and acceptable by regulatory agencies as an approvable composite endpoint to determine positive predictive value. The wrinkle with this subset analysis was then they added on to the PPV, the positive predictive value, the correct localization rate or CLR, and really try to home in on the sort of anatomic specificity of the imaging. Next. So nothing surprising here. It just really shows you that the CLR rate is in line with what you would expect with a positive predictive value. The positive predictive value is a per patient, a whole patient metric. So statistically, it's kind of telling you across the whole patient, did you detect a lesion and was that lesion confirmed to be a true positive based on the composite definition? CLR adds the additional anatomic localization to that. And in some ways, no surprise to see that rate still be pretty high in the high 80% range. Also, no earth-shattering data there to show that on the right, you see, well, what are those CLR rates by PSA level? No surprise, it does go up as the PSA goes up, but you still see pretty high rates even at low PSA levels. And I think that just speaks to the sort of value of PSMA PET imaging. I think we can probably, in the interest of time, this just shows a few different subsets. But essentially, that was a take-home. Pretty high CLR rates, high positive predictive value using the composite definition.
And I think just like one question that I might have here with regards to the PSMA testing, we've seen various different positivity rates. Obviously, some of the protocols vary. There's slight differences from one diagnostic agent to the next. 65% versus 87% in vision. I think we've seen 75%. What are some of those driving factors that you guys see as to the level of PSMA positivity?
Well, yeah, I mean, vision was a very, very, very different patient population, so I wouldn't really see much analogy. I think that the PSA doubling time, the height of the PSA, are things that are particularly important in our practice. I'm sure UCSF has more experience than me, but at the same time, I don't see necessarily a lot of differences between the agents. I think the PSMA 11, gallium 68, and F18 DCF-PYL are pretty comparable from what I've seen. Obviously, one is cyclotron produced, others generator produced. Each has its advantages. I've heard people be pretty dogmatic that one is better than another, but I'll simply say that that's due to two different perspectives. So I see both agents having use going forward. And a lot of what you see in the differences is really just different populations being tested.
Got it. Great. So I think as we move beyond nuclear medicine, it's taken up most of the hour. There was definitely some interesting real world data that was presented. And I believe Dr. Sartor is going to walk us through that.
Yeah. So, one of the things that I think we all understand today is that when you're treating metastatic castrate-sensitive disease, that you have better outcomes if you add in one of the novel hormones. We have unequivocal data from the Latitude study with abiraterone, with Stampede, with abiraterone, new data with Peace-1 with abiraterone, data from Atlas trial with apalutamide, the Enzamet trial, which has survival with enzalutamide. We've got a lot of data now to suggest that adding in a novel hormone to traditional ADT gives good outcomes. And here we take a look at, next slide, please, some real world data. And, it's a lot of patients that were looked at here. And, first of all, they're looking at, who had prostate cancer, and up with 200,000, and then you have metastatic disease at 27,000, and then getting it and making sure they have a health plan and have metastatic disease with kind of confirmation, 16,000, you go on. And you can look and see that you end up with literally thousands of patients being looked at. And then you get these categorized subgroups, and you see ADT only is 2,300, ADT abiraterone 892, et cetera, down the line. And next slide. And what you're seeing basically over time, and you have to realize that docetaxel fits into the equation. I should have mentioned docetaxel because of the data from both the Stampede trials as well as the Charted trial clearly prolongs survival. And yet, the majority of patients in the US that are being examined here are being treated with ADT only. And I'm having a hard time understanding exactly why that is so. The data has been widely disseminated, multiple New England Journal papers, CMEs, etc., have addressed this. I don't know if it's an affordability issue or a refusal issue, or I'm just not quite sure what we're seeing here. But it's a little bit distressing that patients clearly do benefit from ADT plus abiraterone or apalutamide, enzalutamide, docetaxel, yet it's not being used, or it's not being used in the majority of patients. So there's more work to do, I think, with education and maybe more barriers to address with regard to cost. So I don't know, Rahul, what are your thoughts? I'm kind of struggling a little bit to get a full explanation in my mind.
Yeah, likewise. I mean, I think that I'm surprised, on the curve on the left to not see that yellow curve of ADT alone drop more between 2014 and 2019, because that's exactly the period where we saw all the phase 3 study data come out and the FDA approvals change the label for these agents to clearly indicate use in the hormone sensitive setting. So definitely more work need to be done to understand what are the barriers. Probably a deeper dive in terms of the methodology of this particular analysis, just to confirm that it's rigorous and that this in fact reflects reality. But clearly, there are definitely some cost barriers for some patients. Although I'd say in our practice it's a small minority of patients that I think get ADT alone for financial reasons. But our patient population, of course, may not be totally representative of the average prostate cancer patient in the U.S. But clearly more work needing to be done to understand these numbers.
Great, and thank you both. Go ahead, Dr. Sartor, please.
Oh, I was just going to say that when we begin to explore this in more detail, it may need to be very granular, and be very inclusive. You know, I'm practice in a medium-sized metropolitan area. Several practices in a larger metropolitan area. Our experiences may not be representative of those in more rural areas. We're both academicians. We read the literature, we discuss the literature, we have tumor boards. It's a little bit different, perhaps, if you're practicing solo urology and maybe you're practicing in northern Mississippi. There are a lot of issues here. I mean, it would be good to understand it more. The guidelines are now clear, and we do have life-prolonging therapies, and I would hope that as many patients as possible would have the advantage of being treated with these truly life-prolonging therapies.
And so I think in our final few minutes here, it'd be great to hear from each of you on kind of your concluding remarks of, either kind of takes on ASCO or, what you're looking forward to from a development perspective within prostate cancer.
Sure. So, the big take on ASCO, I mean, I did think that VISION was the most impactful study. It clearly has potential to be practice changing, assuming the regulators agree with the data that's been presented. I think they will. If you look at the implications, it's clearly about taking the PSMA lutetium earlier, and we have already designed and already started to enroll big trials that will be looking at the metastatic CRPC pre-taxane space, as well as the upfront patient with castrate-sensitive metastatic cancer. And then, of course, there are a whole other series of trials they're doing. So that's one of my things. We didn't really talk about the BiTE molecules. We didn't talk about the ability for CD3 PSMA bispecific antibodies to engage the immune system. There's definitely some cytokine release syndrome that needs to be dealt with, but these are active agents as well. We didn't really talk about the alpha particles and the development of actinium-based therapies that I think are causing a lot of excitement in some circles. So there's a lot more here, and that doesn't even touch on some of the newer hormonal agents that may be active for those patients who failed Avionine, so clearly we need more data there. So there's a lot to look forward to at next ASCO, including hopefully being in Chicago and being able to see all our friends and have dinner
Absolutely,
together.
Yeah.
I 100% agree with that. I hope we're back in person. I think everyone's had their fill of the virtual meetings. It's just clearly not the same, especially the poster sessions, which are always a lot of fun to get around and get a chance to chat with folks. I think for me, yeah, I agree. Clearly VISION was a big practice changing study for ASCO this year. I think, getting to the real world implications of that, the rollout of lutetium, assuming it gets approved, how will we meet the patient demand for this drug, given how, basically 90% of patients qualify for it in the post one line of chemotherapy setting. I think, there's going to have to be a huge ramp up clearly, both in terms of education and just resourcing and being able to efficiently and treat as many patients as we can to be able to deliver the benefits. I think the rollout's going to be key. And then clearly paths forward with combination therapy with lutetium, other particle emitters such as the alpha emitters, moving the drug earlier. So lots to work on, but this was a really, really important first step to get the phase 3 data out there.
Absolutely. And I think that brings us to a wrap right up on the hour. So again, thank you so much, Dr. Sartor and Agarwal. It's been a great discussion, and I think we all hope that we're doing this in person next year. So with that, everyone can have a great weekend, and hopefully they all enjoyed their ASCO. Take care.
Take care now.
Thank you.
Thank you, John. Thank you, Rahul.